How we study the brain. Lesions and electrical stimulation. Lesions are areas of damage. They map brain functions, and before the 20th century, the only way to puzzle out the function of different brain regions was the lesion method. Today, technology allows us to see exactly which parts of the brain have been damaged in a living patient, but in the past, scientists could learn which part of the brain was damaged only after the patient had died and the brain was removed for examination. Lesions are often caused by strokes or blows to the head, and strokes can disrupt language on the left side of the brain. Today, it is possible to inhibit activity of small patches of the brain temporarily, like a reversible lesion. Transcranial magnetic stimulation is the temporary localized disruption of the functioning of a brain region produced by the application of strong magnetic fields. It uses a coil of wire placed over the skull. Many brain regions are active during any behavior. Stimulating certain areas of the brain makes a person move or hear or see or find something very funny. Scientists can eavesdrop on electrical signals within the brain. Neurons have two types of electrical signals within the brain. The first is tiny fluctuations in voltage around synapses when neurotransmitters bind to receptors, and if enough of these voltage changes, reach the axon. And the second is action potentials that carry information from the neuron's cell body to its axon tips. Devices can detect electrical activity to give us some idea of what's happening in the brain. Electroencephalographs eavesdrop and detect signals under the scalp and it resulted in the modern electroencephalogram, or EEG. The EEG is a recording of fluctuating potentials taking place in the brain made by an electroencephalograph. It reveals that the electrical activity measured from the brain is different if a person is awake or asleep. It can also detect electrical storms during epileptic seizures. In animals, individual neurons are studied. There was a study with a monkey where there are mirror neurons found, and when a monkey watches a human pick up the raisin, as shown in the diagram, then the monkey also picks up the raisin, and a monkey's hand watches the human's hand movement, and the neuron is less responsive if there is no object to be picked up. Sometimes these studies are beneficial to humans too, as shown in the figure. Mirror neurons are neurons that are active when an individual makes a particular movement and when that individual sees another individual make that same movement, so like the monkey. And it seems to provide a neural basis for imitating other individuals' behaviors. There are many different techniques for seeing the brain. The first is a computed tomography, or a CT, which examines the brain structure through computer analysis of X-ray particles passing through the head of several different positions. This is shown in Figure A. Another way of seeing the brain is a magnetic resonance imaging, or an MRI. This uses magnetic fields to generate images revealing structural details in the living brain. It is more detailed and a better sense of varying density of brain tissue than a CT. There are also techniques for watching the brain work. Because if you want to go beyond the brain structure and the brain process, you have to find a way to estimate the brain process, and these are the techniques for watching the processes of the brains. The first is the positron emission tomography, or the PET, a technique for examining brain function using injections of radioactive substances used by the brain. The particle detectors and computers are used to find where most of the markers gather in the brain. The functional MRI, or the fMRI, requires rapidly turning on and off a magnetic field around the whole head. It is magnetic resonance imaging that detects changes in blood flow and oxygen content, identifying regions of the brain that are particularly active during a given task. Psychology and biology are two different vocabularies to describe the same thing of the brain. The brain is constantly changing, and everything leaves a physical change in the brain. The brain machine displays plasticity, or neuroplasticity, which means it can change easily. Our biological understanding of the brain is fairly new and primitive. There are major international efforts to expand our understanding of the biology of the brain. Diffusion tensor imaging, or DTI, is a brain imaging technique that reveals axonal connections.